we are at 3 minutes past, so I would love to take the opportunity to introduce my great friend and colleague, Joshua Welch. I guess we have known each other at this point for almost 10 years. He's one of the Black scientists that every time you read his papers, he goes all the way to the basic building blocks and build the right models together with the methodology to optimize. So it's always very insightful to see different ways of looking at the problem. Today, we have the honor that he's going to present one of the papers regarding self-fate decision, but also looking at the spatial extensions of RNA velocity, which is a very nice follow-up. Previously, we had a talk on self-fate transition using optimal transport, so it's very interesting to compare other ideas as to how dependencies in the spatial context affect self-fate decisions. Without any further ado, I would love to give the presentation to Joshua. Thank you.
Great. Thanks, Shaheen. Thanks for the invitation to speak. I'm excited to talk with you today. So I'll be presenting work that we have out partially in a preprint, and the revised version was just sent in a few weeks ago, now accepted in principle. So hopefully you'll see that paper in a few weeks. So what we've done is develop some computational tools for mapping cell-fate transition using spatial transcriptomics. And before I get to our model, I want to go through some background because we're building on several previous studies that are important to understand first. So the general problem that we're trying to tackle here is the challenge of trying to identify gene expression changes in differentiating cells without serial time series data from each cell. So the ideal experiment that we would want in order to identify gene expression changes in differentiating cells would be something like we put a little tiny camera inside each cell, and we watch from the moment the cell starts its fate transition until it arrives at its differentiated fate. And every point along the way, we would be able to see the gene expression in that cell, and we would have a complete linked time series for each cell on its journey. But what we can actually do with single cell sequencing, for example, is we can freeze the process at a point in time, and cells may be at different points on their journey at that point in time. And we can measure like this vertical slice of the population of differentiating cells. And so this makes it challenging because the cells start their journeys at different times. They take different lengths of time to get to their destination, and they're potentially ending up at different places. And so this is a challenging problem. But one way that people have made progress on this problem is to make some assumptions about what we know about the biochemical mechanisms of gene expression. And so with these assumptions, we can make some progress on this problem of predicting the future state of a cell given its current state and at some future time point. And in my opinion, one of the coolest ways people have done this, approach this problem, is with RNA velocity. And you may be familiar with this concept. The basic idea of RNA velocity is that we see two things about a gene in single-cell RNA-seq. We see the level of unspliced pre-mRNA and the level of mature spliced mRNA. And by looking at the relative ratio between these two quantities, we can figure out whether a gene is likely being induced or repressed at the moment the cell was harvested for sequencing. And the key insight is that you can think about this conversion, this gene expression process, as a conversion between these nascent RNA molecules and mature mRNA molecules, and you can model it as a system of differential equations. And so then if you see a cell that has much more unspliced than spliced counts, then it's likely in the induction phase because the spliced counts haven't caught up with the level of unspliced counts. Whereas if you see a cell in the other phase with more spliced than unspliced transcripts, this is likely because degradation hasn't yet caught up with the end of transcription. Another way of looking at this is there should be a time lag between changes in unspliced transcripts and spliced transcripts. So if you look at a single gene over time, when it's turned on, you should first see U increase, and then after a lag, you'll see S increase. And then when the gene is turned off, you'll first see U go away, and then you'll see S go away. And so you can think of this from multiple perspectives. It's a system of differential equations, it's a constraint on the joint distribution of UNS over time. And either way, it gives you a nice perspective for identifying where a cell is in its differentiation based on the relative amounts of UNS. So the initial RNA velocity paper formalized this idea. They had the really nice insight that this is possible. And then their approach for estimating the parameters of the differential equation system was basically to make a steady state assumption. And this allows basically solving for the splicing rate in closed form using regression. Sorry, the degradation rate. You can estimate it with linear regression. And so then you can predict the RNA velocity for a cell by plugging in its observed UNS counts and calculating the predicted future state of the cell based on the degradation rate for that gene. And this was a landmark paper, but it has a lot of limitations. And Fabian Theis's group pointed out one of these key limitations, which is the steady state assumption is really not a good one to make because we only care about RNA velocity in cells that are not actually at steady state. And so his group showed how you can use all the cells and not just the ones that are at steady state to estimate the rate parameters of this system of differential equations. And the key insight is that if you introduce a latent time variable for each cell, you can do a sort of expectation maximization where you first assign each cell to its most likely time. Then you use those time assignments to take your best guess at the rate parameters. And then you update again the times for each cell using your best guess of the rate parameters. And if you iterate that, then you converge to a joint estimation of a latent time for each cell and rate parameters for each gene. And this was a very nice insight from Theis' lab. Now, a couple of years ago, my lab built on this, and we realized that you could take multi-omic data where you have RNA and chromatin accessibility measured in the same cell, and you can extend this modeling framework further to incorporate chromatin accessibility and gene expression. And so in this case, you can formulate a system of 3 differential equations that involve chromatin accessibility, unspliced pre-mRNA, and spliced mature mRNA levels. And by adding a 3rd differential equation, we get a lot more modeling power. I won't go into all the details. My point here is just to show that this is kind of another step on the way to getting to our spatial velocity model. So again, as with the SC-Velo approach, we can model the triples of C, U, and S across the cells by estimating a number of rate parameters for our differential equation, which in this case tells us things like when chromatin opens and when it closes for each gene, and also the relative timing of when transcription begins and when it ends relative to chromatin accessibility opening and closing. And here again, we used an approach like the SC-Velo dynamical model. Basically, if you assume that there's a normal likelihood that generates data from noise through your differential equation, then you can write out a maximum likelihood estimation for the parameters. And in this case, it's basically just minimizing the mean squared error between your reconstructed data and your original data. And in this case, we had to deal with a couple of challenges, like the 3-dimensional system is not analytically invertible, and so we use a lookup table instead and maintain this lookup table as we train the model. But in the end, we still get this pairing of a latent time for each cell and estimated rate parameters, which allows us to predict velocity and future states of each cell by plugging in the observed C, U, and S counts.
Sorry, quick question regarding the time. So from what I can just tell, the time is inferred to be the same across modalities, right? But we know that some lags exist between, the timing in epigenome versus transcriptome. And so have you seen such differences and lags that we see, especially during differentiation that is known to do the priming first and then observe the transcription after? And B, have you had any chance to extend these in models where you have different times, but also mapping between times of different modalities?
Yes, so this was one of the key themes of our paper, which I won't go into because I want to get to the spatial extension. But Basically, the idea is each modality has a switch time associated with it. So there's a chromatin opening time, which we set to 0 because there's no data observable before chromatin opens. And then there's a transcription start time, which has to happen after the chromatin opening time. And so by estimating those parameters, we can actually calculate for each gene what is the length of time between chromatin opening and transcription initiation, which is precisely the priming interval that you're talking about. And one of the other things which was kind of novel in our paper is we also proposed that there is something called the decoupling interval, which is when chromatin and transcriptional state are out of sync during the repression phase. And people had not really talked about this in the literature before, but we showed some evidence that you can actually see a lag there as well.
Very nice. Thank you.
Thanks. Yeah, and please feel free to jump in with questions. Are people putting questions in the chat also? Okay, yeah, perfect. All right. This taught, the Multi-Velo project taught us a lot about RNA velocity and got us excited about it, but we also realized a lot of the limitations of this iterative latent variable estimation approach. And so we realized that there's a better way to do this by setting up the problem as more of a full Bayesian latent variable model. And it turns out that an elegant way to estimate the parameters of this model is basically through autoencoding variational Bayes. Now, the twist is that instead of just a conventional variational autoencoder that has a fully connected encoder and decoder, the trick in this case is essentially that we swap out the decoder of the VAE with the analytical solution to our differential equation. And there's several different ways you can interpret this. It kind of intuitively makes sense for several different reasons. One way of thinking about it is that we take a VAE and we constrain the decoder so that the latent variables have a physical interpretation. So now our latent variables, instead of just being sort of arbitrary dimensions, they actually correspond to latent time and cell state because of the way that they're used in the decoder. Another way of looking at this is that our generative model is a differential equation, which tells us the constraints on the joint distribution of U and S. And then we simply use that likelihood in order to infer the latent variables through the encoder, which serves as an inference network. So you can think of this approach from those several different intuitive perspectives, but functionally what we do is we have an encoder where we pass in U and S, and then the latent variables in the at the end of the encoder represent a cell state, C, and a time, T. And then we predict cell-specific, gene-specific transcription rates, rho, which we feed into an analytical solution to the differential equation to reconstruct U hat and S hat from C and T. Another intuition that we had is that this latent C is kind of like generalizing the RNA velocity model so that each cell has a cell state. And then the transcription rate parameters are a function of that cell state instead of being constant across all cells, like in the SC-Velo model. So there's a lot of reasons why this makes sense intuitively. And it also has some really nice advantages in terms of giving you distributional estimates. It's much more scalable, and it's more powerful because you're using a neural network to do the inference. And again, this is previous work. We published it in ICML. We have a bioRxiv preprint, which is currently under review. But one of the punchlines is that this allows us to scale up to modeling really large and complex systems where the previous approaches that more or less assume a single differential equation for all of the cells just totally fall flat. So as one example from our preprint, we ran this on the data from Lamano Nature 2021, where they profiled the differentiating mouse brain across time points from E7 to E18. This is a really large data set, and it's got a lot of time points. And one of the things that our approach really takes advantage of is if you have time labels on the data, you can use those as a statistical prior that constrains the likely latent times for each cell. And this helps a lot with the velocity parameter inference also. And so what I'm showing here is a UMAP projection of the 200, almost 300,000 cells colored by a broad cell class. And on the left are future states predicted by VeloVAE, and on the right are future states predicted by SCVelo. And the ones on the left make a lot more sense in terms of what's known about how cells differentiate. And you can also quantify this by calculating a correlation between the true cell capture times and the observed or the predicted cell times for each cell. Okay, so all of this was prior work that we had in our minds, and then we considered all these new emerging spatial transcriptomic technologies, and we wanted to ask the question, how can we take advantage of the spatial information that's in these new emerging technologies and use it to better model cell differentiation? And just to give you a quick review, there's several different classes of spatial transcriptomic technologies. Here we'll mostly be focusing on the first two, which are both sequencing-based, and they perform unbiased transcriptome-wide quantification of gene expression. So one class of approaches performs in situ capture and sequencing, and this would include approaches like Visium HD, Stereo-Seq, low-resolution Visium, and Slide-Seq, and a bunch of other approaches. And these technologies give you unbiased transcriptome-wide measurements, but they require either cell segmentation or deconvolution because they essentially give you gene expression measurements at fixed position spots. And so you have to figure out what's the relationship between cells and spots. Another more recent class of approaches is cell or nuclear labeling and extraction. One approach like this would be Curio Trekker, which is the commercial version of slide tags. And the approach here is basically you melt your tissue onto an array that has spatial barcodes, and then you use the barcodes to label cells in the tissue so that each barcode has a known spatial position. And then you actually pull out the cells or nuclei and perform conventional single-cell droplet sequencing on them. The slide tags Curio Trekker approach works with nuclei because it was developed initially for brain tissue. And so you can do any kind of single nucleus downstream sequencing like 10x RNA or multi-omic or TCR, BCR sequencing, et cetera. And so in this case, the data comes in the form of individual nuclei with known spatial positions, and each molecule can be tracked back to a single nucleus. So you don't have this problem of, does this spot have one cell or two cells or no cells? Every molecule is tracked back to a nucleus. And then, of course, there's a whole class of approaches that use sequential FISH, which are in principle compatible with the kind of questions we want to ask. But in this case, since they're probe-based, you have to pick your targets in advance. And people don't usually pick their probes so that they separately quantify spliced and unspliced transcripts. So we're not going to really pay attention to these technologies in the rest of the talk. So our key idea is to extend the RNA velocity framework into space by modeling an entire tissue as a spatially coupled differential equation. And we built on our previous VeloVAE framework basically by just taking the VAE and replacing the fully encoded layers with graph convolutional layers, either spectral convolution or graph attention. And then we also modified our differential equation framework so that now, instead of treating the cells like they're independent and identically distributed, we treat them as if they are coupled and jointly distributed with a spatial coupling. So the intuition is that instead of thinking about each cell's transcription rate in isolation, we now assume that cell I's transcription rate depends on the transcriptional states of cells J that are near it in the tissue. And you can represent this spatial proximity using a graph that you build based on cell neighborhoods. And then you can learn these functions G that explain how to estimate the transcription rate of a cell given the states of its neighbors. And let me just point out that the way we do this, we're not assuming that cells actually have to have similar or identical transcriptional states if they're neighbors in the tissue, because that often won't be the case. For example, if there's an immune cell right next to a neuron. However, we are assuming, and we're trying to model relationships where your neighbors actually do influence your gene expression. So knowing that there's an immune cell right next to that neuron should tell you something about the parameters of transcription in the neuron. And that's what we're trying to model. And so the basic intuition is we take our VeloVAE network, which has a differential equation solution instead of a decoder. We replace our MLP layers with either spectral graph convolution or graph attention. And then we perform graph autoencoding variational Bayes. So the inputs to the network are actually a graph of cells. The outputs are also a graph of cells. Each cell is a node, and the U and S counts are node features, which we reconstruct. And we don't change the edges, so there's no edge reconstruction.
So we have a quick question from Ziv that is, this seems to be more of a signaling network problem related to these, neighboring cells, not velocity. Isn't that the case? So I would say they're correlated, but I let you obviously answer that one. It is also pasted in the Q&A.
Okay, great. Yeah. So our goal here is to use spatial information to inform our estimation of the transcription rate parameters of differentiating cells. And certainly cell signaling is one mechanism by which cells nearby each other will influence the parameters of other cells. Here we're not incorporating any information about ligand receptor pairs, for example. We're just simply trying to reconstruct the observed spliced and unspliced counts through this differential equation, taking into account the transcriptional states of neighboring cells. So as I'll show later, I think we can get hints of cell signaling underlying these relationships among neighboring cells' transcriptional rates, but we're not explicitly modeling that in this case.
Thank you.
And then, hi, I also had a question.
Sure.
So for this framework, you said the input and output here are graphs, but the decomposition, or at least the equation in B, makes it seem like, we have a graph input and then two scalar outputs for each gene, for each cell, but that the output itself is not a graph. Maybe you can clarify?
Yeah, the output is a graph with node features. The node features are U and S.
Okay, thanks.
Yeah.
And then one thing I wanted to ask, because in the paper it mentions that the framework doesn't take into account things like node features, but it's not mutually exclusive with this framework. It's actually, I think it would be very intuitive to incorporate. But I was wondering if you could comment maybe on what node or what edge features would be in this framework. Maybe
Yes.
I was talking about edge features.
Okay. Yeah, it's a great point. So we did think about this a little bit. Edge features could represent things like, does cell I express a complementary receptor to cell J? And so you could imagine trying to extend this to incorporate some kind of prior knowledge about the mechanisms in which cells could interact. We experimented with that a little bit, but didn't really pursue it very far, just because there's only so much you can do in one paper. But I think it's definitely something worth looking into.
Okay, thanks. And then for context, as you continue to present some of the downstream results, I think an interesting question in all biological modeling is the use of prior knowledge, either for informing the model or for validation of the model. And I get the sense that there's a preference for using this to inform the model and then using things like, maybe reconstruction error or less biased metrics for evaluation of the model. But I'd love to just get your thoughts early on so we can help with results. Yeah.
Evaluation is always challenging in these kinds of settings. So what we've tried to do, you'll see in the subsequent slides, is to come up with kind of orthogonal sources of ground truth that we're not using in inference, and then comparing our results against those. So that's in general what we're doing. And you'll see several examples of that.
Okay, thanks.
All right. So first thing we did was a simulated data study. And here we wanted to basically just ask the simple question, does incorporating spatial information in this way noticeably improve the inference of either velocity vectors or rate parameters? And so to do this, we simulated two different tissue topologies, one where cells are growing outward in a 1D bidirectional fashion, and then a second organization where we have multiple radial axes intersecting. And we picked these two topologies because we see examples like this in the real data that we're analyzing, which I'll show in a second. And basically, we took the previous simulation frameworks from SCVelo and Multi-Velo, and we made the transcription rates and cell states a function of space in the new simulations. And so we have for each position simulated ground truth of the cell time, the velocity vector, and the rate parameter. And we can compare against these when we run inference. And we compared against SCVelo and VeloVAE just to try to see what is the benefit of using a neural network approach versus SCVelo, and what is the benefit of using spatial graph convolution instead of a multi-layer perceptron. And we could see qualitatively that there is an improvement. And we also can see this quantitatively, which I'll show on the next slide. But let me just stop here and point out that these plots look like the stream plots that you see in previous velocity papers, but they're fundamentally different because now the X and Y coordinates actually represent spatial positions. And the arrows actually represent directions of predicted cell differentiation on physical space, which is different from projecting these onto UMAP coordinates. So I'm proud to say we have not a single UMAP plot in this entire paper. Okay, and so once we have the simulated data, we can calculate a bunch of metrics in order to quantify performance. And so I'll just explain these here because we use the same metrics for the real data evaluation. So we calculate reconstruction error, cross-boundary direction correctness, which was a metric developed in a previous paper for quantifying whether you go in the right direction between clusters. Velocity accuracy, which is, I believe, a cosine distance, cosine similarity. And then the correlation between the simulated and measured inferred cell time, and the degree of spatial velocity and spatial time consistency. And we also calculate a k-step version of the CBDIR metric across variable step sizes, because sometimes we see more divergence between methods at a higher step size. Okay, so now I'm going to show you a few examples of how this works on real data. So the first example is from the mouse brain, and this was actually the data set that inspired this project. Initially, this was profiled with Slide-Seq version 2, and they showed that even using SCVelo, you could see some encouraging results, which were that cells seem to be moving from the VZ and SVZ towards the CP. And this actually indicates the known direction of cell migration during this developmental process. And so when we run TopoVelo on this system, it actually really nicely makes a similar prediction that cells are growing and migrating from the VZ towards the outside of the cortex. Another data set that we looked at was Stereo-Seq data. This was published in Cell a couple years ago. E13.5 embryos, and we pulled out the gut and lung. You can ignore this one in the middle. That's an image that shouldn't be there. And the TopoVelo cell vectors predict that the tissue is growing from the inside to the outside. And this is consistent with the structure of the gut and lung, where the gut has these tubular structures that we're seeing in cross-section. So this is predicting the tubes are kind of expanding. And similarly, we're seeing a cross-section through a lung tube, which is also predicted to be expanding outward. And then finally, we tried this on a Visium data set, which was just published in Nature at the end of last year. And this is human pediatric thymus. And in this system, it's known that thymocytes enter the cortex, which is shown in green here, and then migrate into the medulla as they differentiate. So these vectors are predicting the known direction of cell differentiation and migration in the human thymus. So now we did a big benchmarking with TopoVelo and a bunch of other approaches where we ran them on these 4 data sets. And for each data set, we used an annotation of the expected temporal order to evaluate how correctly the direction of differentiation matches what's expected. And for the first 3 data sets, we just did this by hand ourselves based on what's known in the literature. And the reviewers didn't really like that the first time. So we found this data set, which was published in Nature, where the authors had actually used histology to annotate a cortex medulla axis so that each spot actually has a continuous time value, where early time means early differentiation and late time means late differentiation. And in this case, as in the other 3 that we looked at, we found that TopoVelo was really accurately able to reconstruct this expected order. And so we calculated all the same velocity metrics, and TopoVelo consistently performs well on these. Interestingly, we found that the graph attention network seemed to be the best choice across all the settings we profiled, and so we stuck with graph attention. Okay, so what can we do with this approach? So we applied this to another data set. This is a more recent data set from mouse cortex profiled using slide tags. And if you remember, I mentioned that slide tags has this advantage that you extract the nuclei out, and then you know that each read maps back to a single nucleus. And also, since you're performing the sequencing not in situ, the sensitivity of detection is much higher. So this is a really nice data set. And if we zoom in on the cortex, which is one of the most well understood parts of the brain in terms of differentiation, the TopoVelo cell vectors are predicting that cells are differentiating and migrating from the middle of the cortex to the outside. And it's kind of a weird shape because it's in this particular section, it's folded over, but the progenitors are in the middle and the cells are forming cortical layers as they migrate to the outside. And so we wanted to see how far we could actually push these spatial differentiation vectors because we know that cells are migrating in this system. And in fact, people have done really detailed in vivo studies where they perform live cell microscopy and watch these neurons migrate. So there are really accurate estimates of the migration rate of cells in this system. So we wanted to see if we do the unit conversion and actually look at the magnitudes of our predicted velocity vectors, how they stack up against the in vivo measurements. And so to do this, we just need a time interval. And we made the simple assumption, which was made initially in the RNA velocity paper, that the induction repression cycle for genes is 20 hours. And so if we use 20 hours as the length of time that's observed in our slice here, then we can use the known spatial coordinates to serve as the denominator of our calculation. So we have a distance and we have a time, and we can calculate a migration rate by dividing distance by time. So if we do that, the median, which is shown in our box plot here, lines up really nicely with the measurements from in vivo imaging, right around 10 micrometers per hour. And in vivo, they found slight differences between bipolar and multipolar neurons, and ours is within a micron of the multipolar neuron prediction for migration rate. If we plot the velocity magnitudes here, it predicts that the cells that are near the outside of the cortex have the fastest migration rate. We don't really have data to support whether that's completely valid, but it's kind of an interesting prediction. Another thing that we realized as we were analyzing this data set is that since we use the graph attention network, we can actually look at the graph attention weights and try to figure out what the network is learning about the relationship between cell states and neighboring cells' transcription rates. And so to do this, we developed something that we call the cell influence score, which basically adds up the outgoing attention that's being given to each cell by any other cell in the tissue, and then normalizes it appropriately. And so basically this tells us how much influence each cell is exerting on predicting the transcription rates of other cells in the tissue. And what you can see if you plot these cell influence scores spatially is that it's really predicting that a lot of the influence is right here in the middle of the developing cortex region. And this lines up nicely with where the radial glia cell type is. And this makes some sense because radial glia really play an important role in helping cortical neurons differentiate and migrate. And the reason they're called radial glia is that they have these radial processes that neurons migrate along as they develop and migrate through the cortex. And so to dig into this a little further, we quantified ligand receptor signaling using this other tool that we've developed called cytosignal. And cytosignal gives you a per spot estimate of the activity for each signaling interaction across a database of ligand receptor interactions. And so if you add up the number of significant interactions that use a contact-dependent mechanism across all the cells in the tissue, you get this interesting pattern on the right, which seems to correlate pretty nicely with the cells that have a high influence score. And so if you put all the pieces together, what this is suggesting is that the radial glia cells have the strongest spatial influence in the tissue, possibly because of their use of contact-dependent ligand receptor signaling.
Okay.
And just to point out, we didn't build in any information about ligand receptor signaling into our model. This is just something that came out by looking at the weights assigned to each cell in the tissue by our graph attention network.
So quick question regarding the pairs that you used. Are you using the same pairs for all cells, or is there any cell type specificity to these like known interactions?
There's no cell type specificity. Basically, the database says ligand A and receptor B are known to dimerize.
I see.
And so our cytosignal method calculates basically a neighborhood weighted product of ligand and receptor. So it's like for each cell, we multiply L times R in the neighborhood of that cell, and that tells us the likely activity of that interaction in that cell.
I see. Okay. Thank you.
And so the plot on the left colors each cell by the average cell influence score for that cell type. And what I hope you can appreciate here is that this group of cells and this group of cells stand out as having high influence scores. And those two groups of cells are the radial glia and the choroid plexus. And it makes sense that those cells would have a strong influence because, as I mentioned, radial glia are known to participate in signaling to cortical neurons. And the choroid plexus plays a key barrier role. It secretes cerebrospinal fluid, and it creates diffusible signals that influence surrounding cells. Okay. So another thing that we wanted to investigate was whether we could learn anything interesting by applying our approach to a 3D spatial transcriptomic data set. And we found this really nice data set where somebody performed 26 serial sections on an E9 mouse embryo and then did Slide-Seq on each of the sections. And this E9 period in mouse embryonic development is the time when the neural tube is closing, which is a key time in nervous system development. And so we took the beads that were annotated as belonging to the brain and neural tube in this E9 mouse embryo. We extracted out just the brain and neural tube Slide-Seq beads. And then because the mouse embryo has this sort of curved shape, we also unrolled the embryo along the anterior to posterior axis to make it easier to visualize and interpret what the velocity vectors mean in 3 dimensions that are sort of in embryo coordinates rather than in XYZ coordinates. And so to do this, we fit a principal curve through the middle of the neural tube and then unrolled the neural tube along that principal curve. And also, we annotated the regions corresponding to developing forebrain, midbrain, and hindbrain using marker genes, which are shown here. And then we quantified the Slide-Seq data using spliced and unspliced counts and ran TopoVelo on it, and we got this 3D vector field. And so we drilled down a little bit more to try to understand what this 3D vector field was telling us about cell differentiation by using this unrolled mouse embryo. And in particular, one of the most dynamic things that's happening at this point of E9 mouse development is neural tube closure. And it's known that there are 3 different points on the mouse embryo where the neural tube closures start, and then they move toward each other until the closures meet. So there's closure 1, 2, and 3, and the closures 1 and 2 proceed bidirectionally, and closure 3 proceeds unidirectionally from anterior to posterior. And so we were curious to see whether our velocity vectors had any relationship with neural tube closure. And what we found was pretty striking. So we took basically a top view of the unrolled embryo. So this is looking down on the top of the embryo, and the anterior to posterior axis is left to right. And then we calculated the velocity vector component along the anterior to posterior axis, which is the arrows shown here. And then we calculated the divergence of this 1D vector field along the anterior to posterior axis. And what we found is that there are local minima and local maxima in this divergence that actually correspond to where the closures are known to initiate and then intersect. So the local maxima are points where there's maximum divergence, and those are where the closures initiate. And then when closures that are going in opposite directions meet, you have a very negative divergence, which shows up as a local minimum. And so we drew here the positions of the local maxima and minima. And if you compare that with what's known about how neural tube closure happens at E9, it lines up remarkably well. And so it seems that our velocity vectors are actually indicating the directions and origins of neural tube closure in this 3D system. And then finally, we generated our own new in-house data set from a human in vitro model of early development. And we grew these 3D culture system organoids called Embryoid bodies. This is what they look like here under bright field. They're these nice cell spheres. We grew them for 21 days, and then we froze a bunch of them in a single block of ice like this by embedding them in OCT. And so then a cross-section of this ice block will show you a plane that passes through multiple embryoid bodies. And then we took one of those sections, performed H&E staining on it, and then took the next section and performed Curio-Seeker spatial transcriptomics on it. So then we can register the H&E stain section to the Curio-Seeker spatial transcriptomics section. And again, Curio-Seeker gives you transcriptome-wide measurements in 10 micrometer beads over a 9 square millimeter capture area. So this is what the Slide-Seq data looks like. We've got a bunch of spots measuring RNA, and most of them line up with the positions of these EBs. And in order to annotate cell types, we used RCTD to deconvolve these Slide-Seq beads using a previously published data set of embryoid body gene expression. from single cell RNA. So we took the clusters from the single cell RNA data, deconvolved the Slide-Seq data, and were able to identify the expected cell types, including pluripotent cells, early ectoderm, endoderm, endothelial, mesoderm, neural crest, and neurons. And then we also, because we have multiple embryoid bodies in this same spatial transcriptomic section, we also performed spatial clustering using just the spatial coordinates to assign cells to individual bodies. Then once we have the cluster assignments and the EB assignments, we can look for differences among the EBs. And we found that there seemed to be two different types of EBs, one that had more neurons and one that had more mesoderm. And it's not clear if this is sort of two different modes of differentiation for the EBs, or if we're just seeing different parts, different depths of EBs that would be similar if we were seeing them on aligning planes. So then we used the spliced and unspliced counts from our Slide-Seq data to run TopoVelo, and we visualized our cell vectors on the spatial coordinates. And what we saw is really interesting. It predicts that these cells or these bodies are growing and differentiating from the inside out. And you can see in several of these bodies, there's a clear lumen, which is cell-free, and around that lumen are pluripotent and early ectoderm cells. And then as you go further away from the middle, there are more differentiated cells like neurons, endoderm, et cetera. And so we thought this made a lot of sense, and we found some support in the literature from people who did live cell imaging that these EBs do seem to grow from the inside out. We also calculated cell influence scores, and we identified positions within each EB that were predicted to have the largest influence. Interestingly, these don't seem to be in a consistent position, possibly because these EBs have such different shapes. But the cells with the most influence often do seem to be close to this central lumen. And we also found this interesting pattern that there seems to be a strong Wnt signaling activity near these cells that have a high influence. So it could be that these cells have a high influence score because they're also secreting a lot of Wnt ligands. Another thing we realized we could do with the data in this case is actually map out morphological changes because we have H&E data, H&E staining for an adjacent section. And so we took our spatial velocity vectors and overlaid them on the H&E image so that you can see the direction the cells are differentiating and sort of trace the arrows backwards to the cells that are the least differentiated. And what we noticed is the cells that are near the origins of these velocity vectors are more pink. And this is a hematoxylin and eosin stain. Pink, I believe, is hematoxylin, which stains for proteins and endoplasmic reticulum. And so this abundance of pink stain near the origin of differentiation could indicate secretory activity. But in general, the idea of using differentiation inferred from spatial transcriptomics that also has morphology was pretty exciting, we thought. And that's pretty much the data I wanted to show. And I'd like to thank my students, Yi-Chen, Justin, and Jialin, who did the work. And I'd love to discuss and take questions. Thank you.
Thanks a lot. And one quick question I was thinking, slightly out of the context, because you mostly applied it to differentiation, but I was wondering if you had any chance to apply it to slides from tumor samples and see if changes in, velocity can be, which is a interaction between immune and tumor, can be predictor of other phenotypic endpoints, not differentiation, but maybe, for example, treatment resistance in immunotherapy or other type of interaction based between immune and tumor cells?
Yeah, it's a really interesting question. We have not looked at tumor samples at all. I'm not sure it will work. I haven't really looked at any cancer data with our velocity models, but colleagues have told me that the velocity models they've tried haven't worked well on tumors. And it could be just because some of the expectations about, U coming up before S break down when the genetic controls are damaged in tumor cells. But I'd be really interested to try that if you have a data set in mind, or if you want to try that, I'd love to chat.
Ziv also had another question is, can we use these basically changes in velocity as a phenotype and ask what are the proteins and regulators that actually influence these changes in velocity, and what are the again genes, proteins, and regulators that basically drive them?
Yeah, I think this is a really exciting question. I think we did look at this a little bit. I don't think our current model will give very reliable inference of upstream regulators, just because the encoder network is not very sensitive to individual genes. So if you try to do an in silico perturbation, like set the value of some gene to 0 and then pass that through the network, you'll get something almost identical to what you would get if you didn't set that gene to 0. So I think that's a limitation of the VAE loss itself, which the prior kind of makes the encoder less sensitive to inputs in some cases. And so if you want to do that kind of in silico perturbation, you probably would want to tweak the model to make it more sensitive to the key genes that you think are regulators during training.
Yeah. And is there any way, like with other maybe encoders that incorporate, gene networks, or like other architecture, probably you can overcome that because
Yeah,
there's technically not any dependencies on using MLP for
right.
encoders.
Yeah, I think that would be a really, really cool direction to try.
Yeah.
Yeah, thanks for the wonderful talk. So I had a couple of questions. Maybe the one for the most recent slides is on the Wnt signaling. So I was wondering if this was presented based on prior knowledge or systematically identified, that we can see that influence is highly associated with certain genes.
So the way we did it was we basically calculated the cell influence scores shown on the left, and we could see that there were these parts of the embryoids that were being flagged as having high influence. And so then we looked through our list of ligand receptor interactions from cytosignal, and we just asked like which interactions seem to have a similar pattern as the cell influence score. And when we saw Wnt, it made some sense because Wnt signaling is really important for this, embryoid system.
Gotcha. Yeah, and then more generally, how can this tool be used for, let's say, either reducing the barrier to entry for spatial transcriptomics research, or, automated discovery in general? Because I think that, we've expanded from, individual cells and transcriptomics already being a complex and difficult to interpret modality of data to, even more so. Right now, we're trying to understand tissues, and there's lots of bespoke analysis in this paper, right? Unrolling the neural tube to show, the correct velocity across that tube. And so I'm wondering, is it just that you have to be a very strong biologist always, or are there ways to systematically identify this? Because this sort of influence score, I think, in particular, seemed pretty exciting.
Yeah. So ways it can be used, I think some of the really simple insights you can get from it are just like, how is my tissue growing? Which surprisingly can be hard to establish. We've got several collaborations where collaborators were pretty excited when we showed them our predictions, and they're like, wow, this makes sense. Nobody has ever shown this. Let's try to do a validation experiment. So I think just even that simple level of insight is pretty powerful. Yeah, the cell influence scores I think are pretty cool too. I'm hoping that down the line we can actually do something more along the lines of what Ziv asked about, and actually predict how does a perturbation affect the whole tissue. think that that's really exciting. And this is a step in that direction because now we have a dynamical model of how transcription is affected by neighboring cells. And so you can start to ask a question like, what happens if I perturb a neighboring cell? How does that change the transcription rate?
Yeah. Okay. Yeah. So maybe a follow-up to that is that the framework, I mean, looking at spatial transcriptomics across multiple times gives you some understanding of well, you observe the different cell compositions at different time points, and it's not clear at face value whether these are related to differentiation or to migration. So there's a couple examples, like you show the migration of cortical neurons, right? And then you also show the differentiation clearly in the embryos. Maybe I missed it, but is there a way to tease apart these two things in the framework, maybe based on the latent cell state variable? And then two, does it actually matter beyond, like, if you know the composition of your tissue and you can understand it, does it matter if we're looking at migration or differentiation?
Yeah, great question. I think we can't definitively establish whether differentiation, migration, or both is happening just from a single time point of spatial transcriptomic data. I think this is where you might have an advantage if you use an approach like the optimal transport across multiple time points, because there you can actually tease apart change in state versus change in position. Whereas here we only have one time point, and we can predict which direction the cells are differentiating, which may or may not also be the direction that cells are migrating. But without some other information, we can't establish the difference just from a single time point.
Got it.
So I think that in all of the examples where we talked about migration, we were working on other data that told us that the cells migrate in the system.
Got it. Got it. Yeah. So you don't think this would be possible by looking at whether, pseudo time was changing, but cell state was not? Is that not a way you might be able to?
Well, that's a really interesting question. I hadn't thought about it. It just seems intuitively like you can't disentangle the two unless you have real time. But it may be worth thinking about more.
Okay. Yeah. Okay. So then there was on interpreting the plots throughout the study, right? I know we're at time, but so the plots themselves, you said at the beginning, there's no U maps in this. Wonderful. Beautiful. So the way that we interpret the velocity results is actually looking at the gradient of the pseudo time over the image. Is that what I'm understanding right? Or is it actually based on like the velocity parameters?
Yeah, good question. So the arrows come from projecting a velocity in gene dimensional space to real space, physical space. So it's actually a gradient vector in gene expression space that is projected onto physical space.
Okay. Wow. Okay. Super cool. Yeah. So this would also, I guess, relate to pseudo time, but is maybe even like it's a richer representation, just projected onto physical space.
Yeah, pseudo time is more of like an inference based on pairwise distances, whereas and you have to choose a starting point. Whereas here we don't tell it like we think these are early cells. The early cells fall out naturally because they have more unspliced transcripts.
Right. I think I was just thinking in terms of the model, because the model does have this explicit, latent variable for pseudo time. Right. Okay. But this is not explicitly used in the velocity plots. The velocity plots themselves are based on the gene expression velocity vectors.
That's true. Yeah.
Okay. Okay. Thanks.
Yeah.
Okay. Maybe I'll send you some more questions offline. Super interesting. Thank you so much. But I'm going to pause here and let Shaheen wrap up.
So I just want to thank Joshua again for the wonderful talk. As we see still there's a lot of interest in follow-up. We may end up having a platform that people can ask question afterwards and then relate to the authors and presenters. Thanks everyone for attending, and yeah, I will end it up here. Thank you everyone.
Thank you.